Animal cell culture. The introduction. In this video, we are going to learn about the basics of animal cell culture, the requirements and techniques for the establishment of successful cell cultures from the animal tissue, which can be further USALO for cell studies, novel drug researches, lab generation of new organs, and even for new organisms.
Animal cell culture can be described as in vitro maintenance and propagation of animal cells using a suitable nutrient media. Culturing is a process of growing animal cells artificially, although animal cell culture was first successfully undertaken by Ross Harrison in 1907. In 1882, Sidney Ringer developed Ringer's solution, a balanced salt solution of a composition that is close to that of bodily fluids, and successfully kept frog hearts beating after dissection and removal from the body. This is said to be the first instance of in vitro cultivation of animal tissue.
Culture media. Basic environmental requirements for cells to grow optimally are controlled temperature, substrate for cell attachment, and appropriate growth medium and incubator that maintains correct pH and osmolality. Animal cells are cultured using a completely natural media or an artificial media, along with some of the natural products. 1. Natural media. In the early years, the natural media obtained from various biological sources were used: body fluids, plasma, serum, lymph, amniotic fluid, acidic and pleural fluids, aqueous humor from eyes, and insect hemolymph were in common use. These fluids were tested for sterility and toxicity before their utility. Tissue extracts. Among the tissue extracts, chick embryo extract was the most commonly employed. The extracts of liver, spleen, bone marrow, and leukocytes were also used as culture media. Some workers still prefer natural media for organ culture. Synthetic media. Synthetic media are prepared artificially by adding several nutrients, orgtemi, and inorganic, vitamins, salts, O2 and CO2 gas phases, serum proteins, carbohydrates, cofactors, etc. However, different types of synthetic media may be prepared for a variety of cells and tissues to be cultured. It can be prepared for different functions. Synthetic media are of two types. 1. Serum-containing media, media containing serum. 2. Serum-free media, media without serum. Serum-containing media fetal bovine serum is the most common supplement in animal cell culture media. It is used as a low-cost supplement to provide an optimal culture medium. Serum provides carriers or chelators for labile or water-insoluble nutrients, hormones and growth factors, protease inhibitors, and binds and neutralizes toxic moieties. Advantages of serum in culture medium are: 1. Serum binds and neutralizes toxins. 2. Serum contains a complete set of essential growth factors, hormones, attachment and spreading factors, binding and transport proteins. Contents. 3. It contains the protease inhibitors. 4. It increases the buffering capacity. Serum-free media. Synthetic media without serum. Advantages. The limitations associated with the use of serum in the media, described above, are eliminated in the serum-free media. In addition, there are two more distinct advantages. Selection of media with defined composition. The main advantage of serum-free medium is to control growth of the cells as desired, with a well-defined medium. This is in contrast to the use of serum, wherein the growth frequently proceeds in an uncontrolled fashion. Regulation of differentiation. It is possible to use a factor or a set of factors to achieve differentiation of cells with the desired and specialized functions. Disadvantages. Slow cell proliferation. Need for multiple media for different cell lines. Purity of reagents. Development of serum-free media. While designing serum-free media, it is desirable to identify the various serum constituents and their quantities. The most important constituents of natural serum with reference to their use in cell cultures may be categorized as follows. I. Growth regulatory factors, e.g. PDGF, TGF-beta. Ketotine, cell adhesion factors, e.g. vitamins. Ketotine, essential nutrients, e.g. vitamins, metabolites, minerals, fatty acids. Avanines, hormones, e.g. insulin, hydrocortisone. Primary cell culture. Primary cell culture refers to growing and maintaining of the selected cell type, which has been excised from the normal parental tissue. Mechanical or enzymatic methods, enzymatic digestion, are used while excising the tissue. Primary cell culture is of two types. Adherent cells. These cells require an attachment to substrate for growth. Suspension cells. These cells do not require a substrate for growth. They are grown by suspension in an appropriate liquid medium. In order for successful establishment of cell culture, it is important to produce individual cell from group or tissue, and that is called as disaggregation. Disintegration of cells can be achieved by two ways: mechanical disaggregation, enzymatic disaggregation. 1. Mechanical disaggregation. For the disaggregation of soft tissues, e.g. spleen, brain, embryonic liver, soft tumors, mechanical technique is usually employed. This technique basically involves careful chopping or slicing of tissue into pieces and collection of spill-out cells. The cells can be collected by two ways. I. Pressing the tissue pieces through a series of sieves with a gradual reduction in the mesh size. Tertider, forcing the tissue fragments through a syringe and needle. Although mechanical disaggregation involves the risk of cell damage, the procedure is less expensive, quick, and simple. This technique is particularly useful when the availability of the tissue is in plenty and the efficiency of the yield is not very crucial. It must, however, be noted that the viability of cells obtained from mechanical techniques is much lower than the enzymatic technique. 2. Enzymatic disaggregation. Enzymatic disaggregation is mostly used when high recovery of cells is required from a tissue. Disaggregation of embryonic tissues is more efficient with higher yield of cells by use of enzymes. This is due to the presence of less fibrous, connective tissue and extracellular matrix. Enzymatic disaggregation can be carried out by using trypsin, collagenase, or some other enzymes. Disaggregation by trypsin. The term trypsinization is commonly used for disaggregation of tissues by the enzyme trypsin.
Many workers prefer to use crude trypsin rather than pure trypsin for the following reasons. 1. The crude trypsin is more effective due to the presence of other proteases. 2. Cells can tolerate crude trypsin better. 3. The residual activity of crude trypsin can be easily neutralized by the serum of the culture media. When serum-free media are used, a trypsin inhibitor can be used for neutralization. Disaggregation of cells can also be carried out by using pure trypsin, which is less toxic and more specific in its action. The desired tissue is chopped to 2 to 3 mm pieces and then subjected to disaggregation by trypsin. There are two techniques of trypsinization: warm trypsinization and cold trypsinization.
This method is widely used for disaggregation of cells. The chopped tissue is washed with dissection basal salt solution, DBSS, and then transferred to a flask containing warm trypsin, 37 degrees Celsius. The contents are stirred, and at an interval of every 30 minutes, the supernatant containing the dissociated cells can be collected. After removal of trypsin, the cells are dispersed in a suitable medium and preserved, by keeping the vial on ice. The process of addition of fresh trypsin to the tissue pieces, incubation and collection of dissociated cells, at 30 minutes intervals, is carried out for about 4 hours. The disaggregated cells are pooled, counted, appropriately diluted and then incubated. Cold trypsinization, Figure 36.2b. This technique is more appropriately referred to as trypsinization with cold pre-exposure. The risk of damage to the cells by prolonged exposure to trypsin at 37 degrees Celsius, in warm trypsinization, can be minimized in this technique. After chopping and washing, the tissue pieces are kept in a vial, on ice, and soaked with cold trypsin for about 6 to 24 hours. The trypsin is removed and discarded. However, the tissue pieces contain residual trypsin. These tissue pieces in a medium are incubated at 37 degrees Celsius for 20 to 30 minutes. The cells get dispersed by repeated pipettings. The dissociated cells can be counted, appropriately diluted, and then used. The cold trypsinization method usually results in a higher yield of viable cells with an improved survival of cells after 24 hours of incubation. This method does not involve stirring or centrifugation, and can be conveniently adopted in a laboratory. The major limitation of cold trypsinization is that it is not suitable for disaggregation of cells from large quantities of tissues. Disaggregation by collagenase. Collagen is the most abundant structural protein in higher animals. It is mainly present in the extracellular matrix of connective tissue and muscle. The enzyme collagenase, usually a crude one contaminated with nonspecific proteases, can be effectively used for the disaggregation of several tissues. During the employing of collagenase enzyme, the parental tissue is finely chopped with basal salary solution and soaked with collagenase enzyme for 1 to 5 days. After the completion of enzymatic digestion, the individual separated cells were collected through settling or through by centrifugation. Cell lines. Cell line is a permanently established cell culture that will proliferate indefinitely given appropriate fresh medium and space. Cell lines derived from primary cultures have a limited lifespan, i.e., they are finite, see below, and as they are passaged, cells with the highest growth capacity predominate, resulting in a degree of genotypic and phenotypic uniformity in the population. Secondary cell cultures. When a primary culture is subcultured, it becomes known as secondary culture or cell line. Subculture, or passage, refers to the transfer of cells from one culture vessel to another culture vessel. Subculturing or splitting cells is required to periodically provide fresh nutrients and growing space for continuously growing cell lines. The process involves removing the growth media, washing the plate, disassociating the adhered cells, usually enzymatically. Such cultures may be called secondary cultures. Thanks for watching and subscribe to our channel which made science easy to understand.